So in addition to DAF2 and the DAF2 pathway genes age1 and DAF16, there are some other genes that have been well studied in C. elegans and have been shown to affect lifespan as well. And there are a lot of these genes, so we're just going to talk about one set of genes called the clk genes as kind of another example of how genetics can regulate lifespan in C. elegans. And so there are several clk genes. They are mostly called clk for clk. There's clk-1, 2, and 3. We're going to focus on clk-1 gene. The clk genes, or the clk genes, are highly pleiotropic, which means that these genes can regulate a lot of different phenotypes, as we discussed before. clk-1, or clk-1, regulates several different mitochondrial functions. And loss of function mutations in clk-1, or the loss of clk-1 protein function, has been shown to have several phenotypes that might contribute to its longevity phenotype as well, such as a decrease in egg production, a slow development, and a decreased body size. And so if we look at the survival curve here, comparing a wild type and the clk loss of function mutant in blue, you can see that, at least based on these survival curves, the clk-1 mutants have an extended lifespan compared to wild type. And when we look back at some of these other phenotypes, decreased egg production, as well as decreased body size, we might be able to hypothesize as to why those clk mutants have an extended lifespan. So as we already talked about, there's this trade-off between reproduction and the germline and the somatic cells in terms of survival, right? And so the fact that clk-1 loss of function mutants have decreased egg production could actually help contribute to this lifespan extension because more resources are available for the soma to survive and less are focused on the germline. Another possible explanation for this lifespan phenotype is decreases in body size. As we know, small organisms tend to have longer lifespans than larger ones. Additionally, one of the mitochondrial functions that clk-1 or the clk genes regulate is the electron transport chain. And so it's possible that this lifespan extension is also mediated through a decrease in the electron transport chain and a decrease in the production of what are known as free radicals as a result. And when we talk a little bit about cellular aging, we'll talk about how free radicals can harm cells and are thought to contribute to premature aging. And so it's possible that these loss of function of clk-1 also could decrease electron transport chain function in the mitochondria and decrease free radicals and help extend lifespan in C. elegans as a result. But there are honestly a lot of ongoing studies about the clk-1 gene as well as the other clk genes because they haven't completely figured out the mechanism by which it exerts its extension of lifespan phenotype.